Introduction to the immune system for A-level biology. The objectives are: what are the defence mechanisms of the body? How does the body distinguish between its own cells and foreign cells? What is the first line of defence against disease? What is phagocytosis? And what is the role of lysosomes in phagocytosis? So here's an overview of the immune system. So the defence mechanisms of the immune system. So you've got non-specific responses, which can just happen for any pathogen. So first of all, you've got the physical barriers to stop pathogens entering in the first place. If they then get past those physical barriers, the non-specific defence towards a pathogen would be phagocytosis. Then on the other side, you've got more specific responses to specific antigens. You've got the cell-mediated response, or T lymphocytes, and then you've got the humoral response, the B lymphocytes, which is all to do with antibodies. So self and non-self. So in the fetus, lymphocytes will collide with the body's own material, so self material. And lymphocytes have receptors that exactly fit those of their own body cells. But as the fetus develops, the lymphocytes will die or become suppressed, which means you don't keep reacting to self material. So therefore, the only remaining lymphocytes are those that fit foreign material or non-self material, and they will only respond to non-self material. So some non-specific response, some defence mechanisms, some barriers to entry are the ear secretions have an enzyme which inhibits bacterial growth. In the trachea and the bronchi, you've got ciliated epithelial cells which have mucus on them, which will trap any microorganisms, and the cilia will waft the mucus with the microorganisms up and out of the airways. You've got tears, which contain an enzyme called lysosome that inhibits bacterial growth. Tears are also salty, which is a natural antiseptic which dries out bacteria. In the nasal cavity, there's hairs which trap microorganisms. In the stomach contains hydrochloric acid, which kills microorganisms. And then the skin itself is a waterproof barrier. And if the skin gets cut, then platelets in the blood will clot the blood to stop pathogens from entering the blood, or hopefully stop them from entering the blood. So if we look at now, we've done the barriers to defences, we now look at phagocytosis. So the first thing that happens with phagocytosis is chemicals produced by the pathogen actually attract the phagocyte towards them to move them towards the pathogen. Phagocytes attach themselves to the surface of the pathogen. They then engulf the pathogen to form something called a phagosome. So they form like a little capsule with the pathogen inside called a phagosome. Then lysosomes, which contain enzymes, move towards the phagosome and fuse with it. And when they fuse with it, they release hydrolytic enzymes which break down the pathogen, and they hydrolyze larger insoluble molecules into smaller soluble ones. The soluble products are then absorbed into the cytoplasm of the phagocyte. And what the phagocyte can then do is present the antigens on its surface. So the phagocyte will be detected by chemicals released by the pathogen. It then engulfs the pathogen to form a phagosome. Lysosomes will then fuse with the phagosome, releasing hydrolytic enzymes into the phagosome to form a phagolysosome. And then the hydrolytic enzymes will break down the pathogen, and then the soluble products can be absorbed into